Welcome back, guys. We are forging ahead. We are now in the third chapter of the three chapters that are in this first module. And what we're doing is we're kind of building, right? We're going up in that hierarchy of complexity that we discussed way back in chapter 1. We were talking about how chemistry is like this lower level. Within chemistry, we can talk about atoms, smallest unit of matter, that we're going to really discuss. And then we put those together to form molecules and compounds, right? And some of these molecules are real small, like an oxygen O2 molecule. Some of them are water, but still adds up to quite a bit, right? Because we're mostly made of water. And then we go on to really big compounds, like the organic compounds. So whether it's big or small, whether it's inorganic or organic, we're putting all of these molecules and compounds together to form the smallest living unit, which is the cell. All right? So we're going to be in this for a while. I was a cell biologist as a graduate student, and what I hope you come to realize is that the cell, the individual cell, needs everything that you need as a living organism. And really, our organs and our organ systems are really only just to serve the individual cells in our body, right? If we didn't breathe oxygen and breathe away CO2, then the individual cells, millions of them, right, that make us up, wouldn't survive. So we basically have to think about a cell as a living organism almost, and think about how that individual cell gets everything done. All right, so let's start out. We have an image here of a cell of a certain type. Not all cells look the same, but in general we have certain structures that we see across the board, okay? There are three main parts I'm going to talk about here. One is there's a boundary. So again, if we're making this analogy to you as a living organism, we have a boundary too, right? We have the skin, which is a boundary to a whole bunch of things outside of us, and it keeps a whole bunch of things inside of us too. So this is also true of a cell. A cell has a plasma membrane. It has this boundary that's important for keeping certain good things in. It keeps certain bad things out, and there's a regulation of what can go through that boundary, luckily. All right. We have the cytoplasm. The cytoplasm would be the combination of all of the stuff that's inside the cell. Okay. Now, there's a lot of fluid there, and we've discussed how intracellular fluid, mostly water, a lot of it's in the cells, right? 2/3 of our fluids are inside of these cells. But within, we'd see that there's certain structures within this cell. And some of those structures perform certain functions. We call them organelles. So again, in our analogy, we think about the bigger picture of our body. Well, we also have organs inside of us, right? So organelles are doing kind of what organs do for our whole body, right? They're performing specific functions for that cell to survive. And we're going to have to know some of those organelles, what they do to perform certain functions for each cell. Now, technically, cytoplasm, which is everything inside of this cell, even all of these structures that seem to be separate from one another in here, one of those structures would be the nucleus right here, right? The nucleus is one of those organelles. It's just a real important one. It's a control center of the cell. And technically, the nucleus is part of the cytoplasm. But I list it separately here because it plays a real important role as the control center. All right? All right, so first thing that I'd like to do with you guys is we're going to go from outside in, okay? And we're going to focus on the plasma membrane more than anything in our discussions. Why? Because the plasma membrane is very important for regulation of what is kept in the cell, what is kept out of the cell, and it regulates the movement into or out of that cell. Super important things, really important to the survival of that cell, okay, the cell boundary. All right, so if that's so important, we got to understand what composes that membrane. If we understand the molecules, right, we go zoom back now to organic compounds and various other molecules that make up that membrane, and we understand them, then we'll understand the membrane, right? This is the whole deal with our hierarchy. If we understand atoms, we'll better understand compounds. If we understand the whole mix of those molecules and compounds, we'll understand the bigger picture, the cell, okay? So here we are on plasma membrane. So let's talk about something we've already covered, lipids. Okay, lipids is just a fancy name for a fat, right? We determined that lipids, like any organic compound, is made of certain building blocks, right? What we learned before, and we'll put this in blue just for the heck of it, okay? So I'm going to put a little box in blue around these 3 carbons that seem to be joined together by these lines, which are covalent bonds, sharing of electrons, right? And this is glycerol. You're going to need to know that for the test, okay? What are the building blocks of a lipid? Well, glycerol is one part of that, right? And then we see down here that there's a carbon, 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 carbon. I'm going right down the line, okay? I'm going to use orange for this. So all the way down the line here, we see a whole slew of carbons and a bunch of hydrogens that are joined onto a bunch of those carbons. There's a fatty acid, right? That's another building block of a lipid. Now, what we learned before, when it comes to a lipid, is that we could have 3 of these, right? 1, 2, 3 carbons. We could have 1, 2, and there could be a third fatty acid joined to that glycerol to make the typical lipid molecule, or triglyceride, sometimes they'll say. But that's not what we have here. What we have here are 2 fatty acids. Seems that one of them has a double bond here, so it's an unsaturated fatty acid, but that's besides the point. Let's ignore that for the moment. So we have 2 fatty acids. What do we have joined to this last carbon in our glycerol? What we have there, I'll just circle this in purple, is this other combination of atoms that make up a phosphate group, partially. And this phosphate group actually has a charge associated with it. A phosphate has a negative charge. Now, we learned that when it comes to fatty acids, we have all these carbons joined to one another, right? A carbon warring with a carbon for the same electrons. Now, in this tug of war, there will be an equal sharing, non-polar covalent. So down here, hydrophobic. We have these hydrophobic regions. Everything from here down, and here they really just simplify what we're dealing with, 2 fatty acids that are hydrophobic because they have no charge associated, and water does, right? Water has these partial charges on either side. So we have our hydrophobic region of a phospholipid molecule, and we have a charged region, this negative charge that we have associated with the phosphate group, and that would be a hydrophilic region. Now, we're talking about all this. I remind you about all these notions of how a lipid is constructed and how a lipid may or may not want to be dissolved in water. And that's because these lipids they actually are the boundary of every cell, okay? The part of this phospholipid molecule that probably will not want to hang out in water, remember, water exists outside and inside the cell, extracellular and intracellular. There's a lot of it there. So there are certain parts of this phospholipid molecule that will not want to point towards the water. The part that is hydrophobic, the fatty acids. The part that is hydrophilic, the phosphate group. So the phosphate part will point towards water. The fatty acid part will point away. Okay? So here we have another rendition of this, right? This is our early form of what a fat looks like. And here, down here, all we're doing is we're altering that a little bit with this phosphate group. Now these phospholipids will define the boundary of the cell. They make up the plasma membrane. They're the most abundant molecule we find in the plasma membrane. And they're acting almost like a sea of molecules within which other compounds and molecules will be present. Okay? Now I'm gonna talk about this, and then I'm going to show you an image that'll help, I think, to describe this. We describe the plasma membrane as if it was a fluid that is combined of a mixture of different kinds of molecules, mosaic for a mixture of different type things, okay? Different type compounds that are floating in this fluid. We call it a fluid mosaic model when we talk about how this plasma membrane is constructed. This membrane is really thin and is composed of about a whole bunch of these lipids. These phospholipids form a lipid bilayer, we say. And there are a whole bunch of proteins that are mixed within this sea of phospholipids. All right, so let me show you a picture, because a lot of what I just said might have just went right over. And usually if I'm in person with you guys, I'd see that happen, right? I'd see it in your faces. So let me show you a picture now that usually helps this to get across to everybody. I hope it does with you guys as well, okay? So here we have a plasma membrane. This is an image of a plasma membrane. And in yellow, down here, we have the fatty acids. Each one of these little jagged lines pointing down is a fatty acid. And then we have our phosphate group right up above that, okay? And this would be negatively charged. Now, this showing just outside the cell, and we know this is the outside because it says extracellular fluid, okay? And then down below we see where it says intracellular fluid. All right, so now we know that this is indeed a boundary, and that boundary is our plasma membrane between the intracellular fluid and the extracellular fluid. Now, think of how these fatty acids and these phosphate groups would have to interact in order to be comfortable in this water environment, right? Water on the outside, extracellular, water on the inside, intracellular. The only way to have the fatty acids not pointing towards the water, right? We can't have these fatty acids that are basically like oil. They're not going to mix in water, right? We need them pointing towards one another. The fatty acids are hydrophobic. They're going to point towards one another, and then the back of my hand would represent the phosphate group, which is charged. It's okay for charged things to interact with water. We saw that when salt dissolved in water, right? Sodium was charged, cation, chloride was charged, it was negative, right? An anion. And they like to dissolve in water. Well, in this case, that's exactly what we see. We see a phospholipid bilayer, double layer, making up basically an oil boundary between the inside and the outside. Why do I say oil boundary? Remember, these fatty acids are hydrophobic, right? They don't like to hang out in water. I always think about this, I think about my dad when he wore leather boots, okay? He used to put kind of an oil or a wax on his, they used to have a product, mink oil they would call it, and they'd put it on the outside surface of the leather, and that would keep any of the water or salts from, when it's in the middle of the winter, right, from getting into that leather and ruining his boots. Well, this is a similar situation here. We can't have just things that are floating outside of the cell just randomly going into the cell. Those things that are charged can't just come in randomly, because then we wouldn't have control over what's in the living cell. Okay? So this is our sea of phospholipids. Now, you also see scattered throughout this sea these various purple items, and these are proteins. Okay? We have a bilayer constructed of phospholipids, most abundant molecule by far. There's a little bit of cholesterol in there. Cholesterol helps the phospholipids to move, so it is really a moving kind of medium. Okay? That's why we think of it like a fluid. Okay? But then we have these various proteins. Think of the proteins like ships in this sea. Okay? Some of these ships actually have parts that poke out on the outside and poke out on the inside, like you see here and here. They are transmembrane proteins. They go clear through, like transcontinental, right? Or transatlantic, if I go all the way across the Atlantic on a flight. These go all the way across the membrane, so transmembrane. And then we have peripheral. Think about peripheral, like your peripheral vision, right? We're going to only see things off to the sides here. Well, we only see peripheral proteins, like this one here. This peripheral protein only sits on the outside part of the membrane. They're not on the inside. But we have some of those on the inside too. All depends on how they happen to function as to where they might reside. Okay? Now we're going to find that some of these transmembrane proteins actually have a hole in them, and they allow things to go in and out. And some of these proteins are very particular about what sized items are allowed to pass. All right? And some are open all the time, and some are just open when they need to be. Okay? This is regulating what goes in and out. So we think about these as ships in the sea, right? And they're different sizes, so they're mosaic, different size proteins, and they are able to move. Some of them are actually not able to move, and we'll talk about that later, but a lot of them are able to move, so we say fluid mosaic model, talking about this plasma membrane. Okay, the last thing for this clip, talking about the plasma membrane, is let's take a look at proteins. Let's remember, what did we say about proteins? What do we need to remember? We need to remember the name of that group, proteins. We need to remember the elements that make these proteins up. Carbon, hydrogen, oxygen, CHO, right? But CHONS was going to be what we remember for proteins. So nitrogen and sulfur. We need to remember basic functions of the various organic compounds. And in this case, we learned that proteins function in all sorts of ways. Their shape determines their function, and there are lots of different shaped proteins that are in the membrane. So here we have a really broad layout of all the different proteins and types of proteins that exist within the plasma membrane of a cell. Now, some of them I have alluded to already with you, like these two purple ones in the middle. I suggested to you that there are some proteins with holes through them. So here we see a hole, here we see a hole. Some of these are open all the time, like channel proteins are open all the time. They're just a channel through which certain items can move. Some of them are gated. Think of like a gated community, right? It's where only certain individuals are allowed in. So a gated channel would be where certain, not only certain things allowed in, but it's open sometimes and it's closed at other times, right? For a gate would be allowing things in at certain times and not at other times. But then we see all sorts of other functions for transmembrane proteins that are here in this image. Some act as receptors, okay? So think about this, this one way over here, this green one, where we might have a chemical messenger. So let's put it in perspective. Let's say insulin goes out there in the bloodstream. It's going to signal cells to bring glucose in, right? This is our negative feedback we learned about controlling glucose. So insulin has to communicate in some way with cells. So here we have a receptor, and it is just waiting for maybe something like insulin to come along, bind that receptor, and now the cell will do certain things in response to that signal. Okay. Here we see a different one where we're looking at basically an enzyme, right? We knew some proteins can catalyze reactions. All right, some of those enzymes that catalyze reactions are right there in the plasma membrane. So you may have certain of these that can break something down from something big to something small. Okay, let's skip over here where we talked about enzymes. Okay, we talked about transport, things getting in and out. Cell recognition. All right, so some of the proteins, like we see over here, like with this orange one over here, this one is a cell identity marker. So my identity, right? You know me from all my videos so far, got kind of dirty blonde hair, maybe it doesn't look that way, going gray, by the way, skinny guy, you know, whatever. Those are ways that you identify me. Your immune system identifies your individual cells as well, okay? It identifies you as being, these cells as being normal. It will maybe see viruses and bacteria as abnormal, so it wouldn't recognize those. So cell recognition is an important function of proteins. And even when we talk about cells, some cells have to stick to one another. Like my skin cells all stick to one another, so these cells don't just fall apart if they get a little friction, right? A little bit of abrasion. Some cells don't have those kinds of attachments, like red blood cells generally don't. They're not meant to stick to one another when things are going normally. But this would be some form of cell adhesion. Okay, we'd have a cell adhesion molecule for attachment to one another, like you see here, or maybe cell attachment to some kind of other proteins that exist underneath the skin to keep your skin from bubbling right off and peeling off, right? So lots of different protein functions. What we're going to do when we tune in next time is we're going to still stay on this plasma membrane. We're going to be on this plasma membrane for a good 2 or 3 clips because it's that important to understand the regulation of these things, okay?